I said, welcome to class. This is CRP305, and I still remain your facilitator. Today, we will start with tri-hybrid cross. I'm going to start with tri-hybrid cross, and like you all know by now what a monohybrid cross is. Monohybrid cross involves one trait, a single pair with two alleles, a single pair of gene with two alleles. It is for dihybrid, you have two pairs of genes, four alleles. Explain what these things are. I want to refresh your memory. You can always go and listen to our the recording is on your platform there. And listen as many times as possible. So with dihybrid cross, you have two pairs of genes with four alleles. So for a trihybrid, tri means three. Trihybrid cross, you have three pairs of genes. And definitely you have six alleles involved. Like you can see here, these are the parents. You have the dominant gene here, another one. So the gametes that will be produced here, you have to know the difference between all these things. That's why I took my time to explain the terminology, so that as we are going on, when I talk, I expect you to understand by now. The gametes, this is the gamete produced here. This is the gamete produced here. So for the F1 generation, when they cross, this is what you get in the F1. Some of you are making mistakes. For gametes, I told you, if you want to write the gamete, you are going to choose one of these that is appearing twice. Choose one each. This is double A. You choose one for the gamete. This is double B. Choose one. Double C. Choose one. That is how you do. That is how you write gametes. When I was telling some of you to write the gametes, I was saying all manner of things. All you just have to do is just to select those that are appearing twice. If it is not appearing twice, you don't select one. So these are the gametes produced for the first parent, the gametes produced for the second parent, and this is what you have in your F1. When both of them are crossed or when they fertilize, this is what you have in your F1. And when to get your F1, what do you do? What can somebody should tell me? Let me choose. Okay, Umar, what do you do if I say you should get the F2? What do you do? F2. I can't hear you. You have to mute your mic. Mute your mic. How do you get the F2? The F2 generation.
How do I get the F2? And then I will use the Phoenix square with this.
What do they call it?
I will self the.
That's it. Self. That is what I want to hear. You self F1. To get F2, you self F1. To get F3, you self F2. To get F4, you self F3. That is how you get your generation, your filial generations. You do it by selfing. So you self. Like say you should get the F2 for me, you have to self. So to get the F2 of this F1 generation now, you self. That means you mate it to yourself. That's the meaning. We are saying that you are selfing something, you are mating it to itself. So this is how it is done. So in this case now, the gametes that will be produced, some of you are making mistakes too, but you can use a square to do this. You can use a square to do this. I'm not going to teach you because you can use a Punnett square. Okay, that's that box that you normally use. Some of you used it in your F2. You can also use it for this one. It will be neater. Because if you start using all these lines, it's going to be rough. But there's a technique. Some of you got it in the, and I told you that it was correct for your assignment. So it's the same thing you will do here. You are going to pair it up. The garments that are produced, you put it horizontally, the same one again, you arrange it vertically. And then you start to kind of do like a multiplication to get the your phenotype and your genotype. So there's a formula for getting the number of your gametes, to make it easier for you. If I tell you what is the number of gametes produced in so cross, you have a formula for it, and the formula is 2n, where the n is equal to the number of the heterozygous loci. So in the case of a dihybrid cross now, your n is 2 because it's dihybrid. In the case of a trihybrid cross, your n is 3. So it is 2n. Okay, that is the formula for getting your gametes. And yes, that n is raised to power. Mistake here, it's raised to power. This should be a superscript. So same thing for a trihybrid. Let's consider three heterozygous loci. So the number of gametes is 2 raised to power 3. This is raised to power, not 23. Raised to power 3. 2 raised to power 3 is the same thing as 2 times 2 times 2, which is 8 gametes. So 8 gametes is formed. For this trihybrid cross that you want to do now, you are going to get 8 different gametes when you self this. So it's the 8 gametes that you will arrange horizontally and vertically, and then you will use them for your Punnett square. So I'm giving you a hint, and you construct your Punnett square, and then I want you to attempt it. I want to see. So this is a table here for the formula that I talked about. And in the case of that trihybrid cross we are talking about, these are the gametes that will be produced. I've even written it here for you. These are the 8 gametes that will be produced. Capital A, B, C, capital A, B, small c, capital A, small b. Different combinations. You can see that they are in different combinations. So these are the 8 gametes that will be produced in your trihybrid cross. So is this 8 that you will arrange in your, let me show you on the whiteboard, your Punnett square now. You are going to draw a square like this. You can see my board. So, and you will put it in sections like that. Some of you did it in your assignments. You are going to put it in sections like that. So the first one, what you have here is your capital A, B, C. The next square, you have your just showing you so that you know what to do in your assignments. Capital A, think small b and capital C. 8 gametes that are produced have to everything in your everything in your square, your unit square. Capital, capital, small. Change this. You can see it. You are going to list all the 8 gametes here. These are the 8 gametes that are produced, and you can see that they are not the same thing. They are not the same, they are different. And I told you that anytime you want to you are going to list the 8 here. You will do them horizontally like this, and you will do them vertically on this side as well. Then you will start to, for example, now, let me just do the first one for you. To lose too much of our time. So you have the same here and here. You arrange the other ones too down here. 8 of them here. So you will now start to kind of do like a multiplication. So this first box here now, because they are all capital here and all capital here, so you are going to get your result here as capital A, capital A, capital B, capital B, capital C, capital C. This is what you have in the first column here. So you are going to do the same thing. You will do it for the 8 all through this place. Then you will now start to count which one is the phenotypic and genotypic ratio. So I think you are able to do that. Able to do that. If you understand, is there any question? Okay. Let's continue. This is your assignment. This is your assignment. I've already done the something for you. All you need to do is just to self find the F2 generation. This is the F1, and it is self here. So you have to find how many gametes we expect and the genotype. So you use the Punnett square to get the expected genotypes and their ratio. This is another assignment. Please attempt it. So we talked about the Mendel principle, the Mendelian principle, and for the monohybrid cross, who can remind me what is the phenotypic ratio for monohybrid cross? Phenotypic ratio for monohybrid cross. So you don't forget.
3:1.
3:1, yes. Okay, somebody has answered already. Okay, that is the phenotypic ratio. And what about for the dihybrid cross? What is the phenotypic ratio for dihybrid cross? And tell us.
1:3 to 3:1.
9:3 to 3:1. Okay. So that is the expected thing. That is what you expect when you do a Mendelian cross. That is the expectation. But in some cases, because of what genetics is, in some cases it is not so. There's always a deviation in some cases. There's a deviation from the normal Mendelian ratio that you are used to. The 3:1 ratio for monohybrid crosses, the 9:3 to 3:1 ratio for dihybrid crosses, and for trihybrid crosses, and so on and so forth. There is a normal thing, and it does not happen like that in all cases. Sometimes there is a deviation, just like we have in human beings also. Okay, you know, there are some people that are called special people. They have what we call Down syndrome and so. So they are not in quotes normal as in terms of human beings, because the normal human being has what you call 23 chromosomes. Okay, a normal human being has 23 chromosomes, but in the case of these people, they have more than 23 chromosomes. Okay, so they are special people, and this kind of thing too happens in crops. This kind of thing happens in crops. You see a normal crop, it's like for instance, maize. See a normal maize growing just one straight like that, the stalk straight, and has maybe like 2 or 3 cobs. In some cases, you see maize that is not normal. That you even know that this maize, mere looking at it, you know that it's not normal. So there are some reasons for this deviation from this ratio that we'll talk about. And some of the reasons, 3 of the reasons are stated here. The first one is the pleiotrophy. Okay. Second one is linkage. And the third one is epistasis. So these are the some of the reasons why there's a deviation from the normal Mendelian principle. So if there's a deviation, that is why chi-square is involved, because chi-square is a test. It's a test is a valuable tool, okay, that aids a farmer or an investigator to determine the goodness of fit. There's something that we call goodness of fit in statistics. So this chi-square is the one that will make you to know the goodness of fit, and it's important for genetics, especially when you do your field work and your statistical analysis. So plant breeders use chi-square mostly when dealing with qualitative traits. We'll talk about that later in the day. Qualitative traits that you cannot measure, that are not measurable, like your leaf color, your flower color. These are traits that you can see, but they are not measurable. And these traits are not usually affected by the environment, okay? So it is mainly a genetic thing. So, well, like I said, there's a segregation ratio, 3:1, 93 to 3:1. When you plant 100 crops, for instance, that you are a geneticist, you should know that this is the ratio that you should expect your phenotype to be. But when you go to the field and you don't see that ratio, you now begin to wonder what is happening. And so when you want to do your chi-square, you have to subject your plants to chi-square tests. And like I told you in the previous class, we are going to do mathematics today, but it's going to be easy. You don't have to be worried. It's not something that is too difficult. I'll try and explain and then give you assignments, because things like this, you cannot, but you can't escape it in the exam, as in you have to know it. You have to know it. So we are going to be talking about so we'll be talking chi-square, and there's a formula for chi-square. You can see my screen. Okay, somebody said I should show the slides to the assignment again. Okay.
I will send it to the group. I already screenshot it.
Oh, that's perfect. That's why it's good to have competent class rep. She said she will send it to the group. Well done. Thank you for that. Okay. So I hope you can see my screen now.
Yes, ma'am.
So, like I said, in a monohybrid cross, the normal ratio that you expect in your F2 population is the 3:1 ratio. In a dihybrid cross, the ratio that you expect is the 9:3:1 ratio. 9 for the homozygous allele, 3:3 for the heterozygous, and this one for the homozygous recessive allele. So in the trihybrid cross, the reason why I put that here is that I give you an assignment and I expect you to find the phenotypic ratio. Okay? How do you now use your chi-square? This is the formula that you need. This X2 is chi-square, is the symbol for chi-square. Okay, and it states that chi-square is equal to your observed value minus expected value squared. That means observed minus expected squared. You can see the brackets, and the square is at the back, all over expected. Okay, where your n, this is a summation sign, this is an addition sign for those of you that are used to mathematics. Your n is the observation, the last observation. I is the first observation. From this one is not really important, but this O, just know that is your observed value. E is your expected value. Okay, this sign, like I said, is the addition. So you have a bigger one here. This is observed minus expected, all over expected here, squared. This is your formula for your chi-square. It's simple, you should know it. It's easy to know like that. And then I want to give an example. So I already explained this. Data observed on the field from the segregated ratio may not be perfect, like I explained before. You have some deviations, and I told you the reason why. I mentioned 3 reasons why you have the deviation from the normal Mendelian ratio. So we want to work with an example now. And the example says, consider a monohybrid cross of yellow and white cotyledon. So there's a plant that has yellow cotyledon and white cotyledon. So in this case now, the F1 are all yellow. This simply means that the yellow is dominant over the white. So the F1 is all yellow. And in the F2, however, you know they have said that is a monohybrid cross. So you expect that in the F2, you get 3:1 ratio of yellow and white. So the assumption here is that we planted 1,000 F2 plants. And since it's a monohybrid cross, the segregation ratio is expected to be 3:1. That means 750 for the yellow cotyledon and 250 for the white cotyledon. That is how you do your ratio 3:1, right? But in this case, on the field, when you got there, expecting to see this 3:1 ratio, what you got is 740 yellow and 260 white. You know that it is different from the normal Mendelian ratio that you are used to. So the question now is that, can you accept it? Can you accept this result as good enough? Or you have to reject the whole experiment and start again. So that is why the chi-square is important. You want to know, how fit is this result? Can you accept it, or is it rubbish that you have to start again? That is why you have to do this chi-square. So the first thing that you do in this case is that you write your hypothesis. The hypothesis, for those of you that are used to statistics, there's anytime you want to do any kind of calculation of this form, you have to start first with your hypothesis. And in this case, the null hypothesis is the observed value is equal to the expected value. Is either, as a scientist, is either your experiment is positive or negative. If you have a negative result, that doesn't mean that your experiment is bad. You have to report it that way. In some cases, that is even what people are looking for. It's not every time you have to get positive results when you go to the field. If you do things the way you're supposed to do it and you still get a negative result, you report it that way. It can be for several reasons. So your observed is equal to expected is your null hypothesis, and the alternate hypothesis means that your observed value is not equal to the expected value. So this is how you write your hypothesis. So I want you to just follow me carefully and take notes. If you have any questions, you ask. Still going back to that question, in a monohybrid cross, we have the ratio 3:1. That's 2 classes, 3:1 ratio, right? So the first thing you will do now is to add the ratio 3:1. When you add 3 plus 1, you get 4. Okay? So you add your ratio 3:1, you get 4. So when you now add your ratio, you know that your total plant is 1,000. So all you have to do is that you take your total plant and you divide it by the ratio, which I have done here. 1,000 divided by 4, and you get 250. Okay, I hope it is clear up to that point. Is it clear to that point?
Yes, ma'am.
Okay, so you get your ratio, you add your ratio together, you get your 4, because it is a monohybrid cross. Of course, you know that if it is a dihybrid cross, you it is not 3 to 4 that you are going to add. But in this case, this is a monohybrid crop, so you're adding 3 plus 1 to get your 4. So you get the total number of crops. That one is always, they will give you that. Okay, you have to know the total, and in this case, the total is 1,000. So you divide your 1,000 by the ratio that you added, you get your 350. So the first class now, you know, I said there are 2 classes. There's a class of 3, there's a class of 1. So for the first class, the ratio that we'll be expecting, you know, in the question, they have already given you your observed value. The question, they have given you your observed. So this is your expected now that we are calculating. You will always calculate your expected, your observed will be given to you. So in the first class of 3 now, your ratio now will be 3 for your ratio times this 250 that you just got by dividing 1,000 by 4. So you now get 750. That's for the first class. And for the second class, which is the ratio of 1, you have 1 times 250. Get your 250. So these 750 and 250 are your expected values. So now you form a table, you put expected on one side, you put observed on the other side. Like I told you, this expected is the one we calculated, and this observed is in the question already. So for the yellow cotyledon, you just put in your value. Yellow cotyledon, the expected is 750, the observed is 740. For the white cotyledon, the expected is 250, while the observed, which is given your question, is 260. And it's very important, when you add, you must get the total number of plants, which in this case is 1,000. So when you add, to know that you are on track, you have to get the total number of plants. Now, using your chi-square formula, which I have given you before, you have your observed minus expected all over expected, which is here. You just substitute, and then in this case, you get minus 10. If you multiply, if you get the minus 10, if you square minus 10, you get a whole value, because you can't get negative value, so you have to be careful. You square it, you get your positive value. So you do that for the first class, and you get your answer 0.13. Then you do it also for the second class, which is the ratio of 1, and you get your 0.4. I hope it's clear to that point. So you have to do an addition, which is the summation of the 2 values that you got, 0.13 plus 0.4 to get your 0.53. So your calculated chi-square value is 0.53. So that is for the first step. That is how you get your chi-square value calculated. Okay, so there's a chi-square table, like your 4-figure table that you know. If you have your 4-figure table, you should be able to check. But in the case of this one now, in your study material, your coursework, there's a chi-square table there. There's a chi-square table there, and from there, you will go and check your chi-square tabulated. This one that we did is the calculated. You will now go and check the one on the table. The one on the table is tabulated. Okay, and your degree of freedom. Let me see if I can get your chi-square table from your material here, so that I will show you. Even though in the exam, I won't encourage you to do your table. I may give you the value, the tabulated value, okay, so that you can use it to compare. But I just want to show you what it looks like. So, let's see if I can share. This is what your chi-square table looks like. This is what it looks like. So your degree of freedom, in this case, because there are two ratios, you are working with two ratios. So your degree of freedom will be n minus 1, which is 2 minus 1. N is 2. The degree of freedom you'll be using here is 1. So everything you check, you are checking under this 1 here. Hope you can see 1 on the table. Can you see the chi-square table?
Yes, ma'am, can see.
Yes, you
You
can
can
see.
see.
Yeah.
So everything you'll be checking is under this 1 here, because the degree of freedom is 1. Now, the value that I was talking about is the calculated value is 0.53. We're going to check where does 0.53 fall under this. Somewhere between here. So I said I'll give you your tabulated value. 0.5 somewhere. Check better. You understand it to this level, right?
There's only 0.55.
Okay. 0.55.
Understand this table. I don't know how to check.
Number 5.
Number 5, right?
Yes. No, vertically number 5. Yeah, after 0.41, 0.5.
Around this place. 0.55. I will check back. I'll check back. Let's just continue. Let's continue. So oh, okay. Degree of freedom at 0.05 level of probability. Okay. Okay. 1 degree of freedom at 0. Okay. Level of probability that we usually use in agric is 0.05. Just simply means that for every 5 plants out of 100, you should get the same result. In medicine, they usually use 0.01 because we are dealing with lives, but in plants, okay, I think that is where I was confused. This is 0.05 here. But anyways, let's just continue. Let's continue. Okay. Like I said, you know how to get your degree of freedom. And when they checked the table, they got 3.84 as the tabulated value. Tabulated value, and this 0.53 is your calculated. Okay. Like I said, I will provide you this value for your table. I'm going to provide you this value because I won't encourage you to bring any form of paper or anything to the exam. So I'm going to provide you with this table, this value for your table. So what you have to do is to compare 0.53, this value here, compare it to this value on the table. Okay, that's what you have to do. And he said, or in chi-square, when you compare two values, calculated value must be greater than the tabulated value. Must that I said does not mean that it should be like that. What I'm trying to say, if the calculated is greater than the tabulated, then you accept it. It is significant. That means you can accept that kind of result as a Mendelian principle. But if it is the other way around, if the tabulated is less than the calculated, then it is not significant. So you cannot accept that kind. It's not good enough. It's not fit to be accepted as a Mendelian ratio. So in this case now, it was not significant. So the observation, okay, sorry, I'm mixing something up. If it is not significant, then you can accept. But if it is significant, just what I want you to do in this assignment now is that try it. This is another monohybrid cross. You have your observed value, you have your calculated value. You can, or maybe we should just do this in class and then let me do this as an assignment. So this is a question here. A breeder made a monohybrid cross and is expected to see a phenotypic ratio of 3:1. He observed 215 plants to be resistant and 85 plants to be susceptible out of 300 plants used. Use chi-square to prove what he observed and what he expected to see. So draw up a hypothesis and valid conclusion. So in this case now, they are talking about disease resistance and susceptibility. So your hypothesis now will be, who can help us with the hypothesis? Your null hypothesis will be what now? Let's try this out. Moving to my whiteboard. And help us. you don't talk, you will go and do it though. Just go and do it and submit. Because nobody is talking now. I'm not going to say anything. I've already explained my own. Nobody is talking now. So if you don't say anything, then go and do it and then submit as assignment. I want to know if you know it. That's why I'm asking questions. What's going to be your null hypothesis now? We are talking about plants that are resistant to disease and plants that are susceptible to disease. So your null hypothesis will just be that the plants that are susceptible and plants that are resistant are the same. Or it is the plants that are susceptible is equal to plants that are resistant. And the alternate hypothesis will mean that the plants that are resistant is not equal to the plants that are susceptible. It's just simple like that. So you just say resistant plants is equal to susceptible ones. And for the alternate hypothesis, say that resistant plants is not equal to that sign of equal to with a slash on it. Okay, it means not equal to. That's what it means. It means not equal to. When you do your equal like that, then it means not equal to. It is not equal to susceptible plants. That's what your hypothesis will look like. So what's the next thing we will do? You have got your hypothesis now. So what is the next thing you will do?
Apply the formula.
What's the formula?
The formula is S squared.
Before you apply the formula, what will you do? If you go back to the question, you see that you have your observed value. So you first of all look for your expected value before you use the formula. So you know how to get your expected value. I've already explained it. You know your observed value. So just put it inside and let me see, because it's 11 o'clock already. Do it the way I explained it and let me see the result. Any question?
Please,
Yes.
Hello, ma.
Yes, I'm listening.
My question is not directly on this one. I want to ask about the exam. Will it be theory and objectives?
Yes.
Okay.
No, it's not. It's going to be only theory. You are in 300 level now.
Okay.
It's theory throughout.
Okay.
The only objective is your TMA. So this one is theory now.
Okay, madam. I want to say this. The group created, I can see maybe my name is not included.
Yeah.
Hello, ma.
Yes,
Drop your
I can hear you.
I dropped it.
Your name is not included where?
The group, this WhatsApp group for this.
Okay,
course.
okay. Talk to your class captain now.
Okay.
Gloria,
Maybe
there,
she
somebody
can
said her name is
drop her own number so that we.
All right.
Please drop
Let me
your
draw my
number.
number.
Okay.
So that is the mathematics for you. It's not difficult. Not difficult. No, you people are in university now. I shouldn't be spoon feeding you like that. I've already explained one to you, so just go and try this out. Let me see what you can do. You know that this is a monohybrid cross. The phenotypic ratio is 3:1. So
Please
you
drop the
add
number
it.
again.
You said?
She has not dropped the number. Sorry, ma'am.
I've dropped
Drop
it.
your
Check the
number.
group chat. I've dropped my number already.
Drop it inside this chat, this place, so that they can see it. He will not be able to see
The
it on
number
your
is
group
there
chat.
already. The number is there already.
On the chat?
Yes.
Yeah,
can't
check
see
your
it
chat
now.
there. Should be there.
It should be there.
So just try it out. Get the calculated value and submit to me. So there are two assignments here.
I have a question, please, ma'am.
Right.
I have a plea. This PowerPoint, please, ma'am. Can you drop it on our course page?
On your course page.
Yes.
Recording is there.
Hmm?
The recording is there. Facilitation. Your recording is on the facilitation page.
Okay.
It's there. Check it. If you can't see it, let me know. So before you close, let's quickly talk about qualitative and quantitative traits. And quantitative traits, quantity, just like quantity, all right? These traits are, they are measurable, and all aspects of an organism's morphology, physiology, behavior, fitness, talking about plant height, things that you can measure, plant height, number of leaves, number of branches, those ones are quantitative traits. Or in the case of qualitative, those ones you cannot measure them, like your fruit color, your flower color, and things that have to do with things you cannot measure, okay? And there are differences between your qualitative and quantitative traits. There are 5 differences here. In the case of qualitative, you have discontinuous variation, and for the qualitative, we have discontinuous variation. For quantitative, we have a continuous variation, like your body weight, grain yield, and so on. For the qualitative traits, is governed by a single gene or few genes, while for the quantitative, it is governed by many genes. And these are things you can read up by yourself, concerned with individual mating and their progenies. In the case of quantitative, you have it's concerned with the population and all kinds of mating. So there are 5 differences here. If you must know, you have to know the differences so that you'll be accustomed with it. So I think I can comfortably enter this class. Any other question again? Like I said, there's
Yes,
no
ma'am.
sin
Yes, ma.
I
in not knowing, but you people have to meet and explain to each other how it goes. Yes, what's the question, please?
please, my question concerns our TMA. The question set in our TMA, the options are not the same. The option, there's no correct option. And when we choose anything, we just choose and we get low mark. So some of the question is not corresponding with the course material.
Really? TMA?
Yes.
Yes, ma. Yes.
Yes.
Send it to you, but I was like, I don't want to stop you. So I wanted to
TMA.
that's that.
It's
Yes,
not
ma.
in the course. Like which
The
question? Can
option
you
are not
remember
tally
any
with the
question?
question.
Options do not tally.
Please,
Yes.
can I forward the question to you on the WhatsApp?
Let me see it. Let me see what you mean, because we always send our questions to DEA. They are the ones that used to upload and do all these things. So I'm surprised at what you are saying right now, because
I'll
we
send it to
send the questions to DEA. DEA is the one that will upload it. So if they are not uploading it correctly, then that's a problem. That's a serious issue.
Sorry,
Your
let me come.
TMA is
Glory,
supposed to make
Glory.
30%. Yes.
Yes.
Please, just forward the question on the WhatsApp platform so we have a look at it, because there is things you don't maybe you are not conversant with and you're supposed to. My TMA, I finished everything, but the TMA was really
If
good.
somebody,
but
if somebody is saying that
let's have the
he
question.
did it,
Let's see.
if somebody is saying that he did it, then
happening is that
there's no
we are not
problem.
having the same question. They are scattering questions for us. Your question may not be the same with my own. That is what is happening.
Let's have the question. I'm not confronting you, Glory. Let's have the question. Let's see the question first.
Hello.
Yes,
Now
Hello.
is the question.
Hello,
Yes,
ma.
Let's see the question first.
Hello, ma. Yes, ma. Hello, ma.
Let me see your hand up. Yes.
What happens is that the option given in the question are not tally with the question itself. As in, for instance, they're asking you what is the color of the clothes, and meanwhile the answer is supposed to be black. They will be giving you an option like the house is green, the house is see something that is not tally with the that does not relate with the question at all. That's what they're giving in the option.
Like I said, let me see the question. Hey, Doku, your hand is up. Yes.
Okay. Yes, ma. Good morning, ma. I want to ask about this gene. Now, you said the gene is either homozygous or heterozygous. I want to ask which one is the best, or what is the advantage of one to the other?
You know that plants have different uses. Now, for example, let's talk about maize. Maize, the economic part of the maize is that cob. That's the leaf is not really useful to us. The stalk is not all what you want as a farmer is that you harvest your maize. And when you open the cob, when you open the maize plant, you want to see a full cob. When you open the ear, you want to see a full cob. See something that is full, right? So this homozygosity and heterozygosity of the thing, it differs because somebody is talking, please mute your mic so I will disturb our voice. So what that means is that in the case of maize now, sometimes if you want yield, like what we are talking about, the ear must be full. You want it good yield, and you can see that yield in one parent. You can see it somewhere. Maybe you see one maize and you say, this maize is very big, it has full cob. But as it is like that, you plant it, before you know it, insects are all over the whole place. You know, anything that you like is what insects you like. So it is easily a disease or whatever, it's easily eats it up. And you see another plant somewhere that the cob is not doing well at all. The yield is not good at all, but insects will not go near it. Now, what you want is to get that gene that is inside that one that is not making insect to pursue it, and get the other gene that is making it high. So you now cross it together. One, in the case of that yield, it is homozygous for yield. It does very well. but it's recessive for insects. What do you have? And the other way is the other way around. So when you combine these genes, depending on the crop, sometimes it's even as a farmer, is the recessive, that one that is scarce that you don't see at all, is that one that the farmer is looking for. Depending on the crop, sometimes that recessive one, that one that comes only once, is the one the farmer is looking for, and it's very difficult to get. Sometimes it is the one that is dominant, that the farmer does not even want. So it depends on the crop you are working with. There's nothing like one is better than one. You as a farmer, what is it that you're looking for in your plants? Is it on the recessive side? Is it on the dominant side? So these things differ. That's why I don't really know how to answer your question. The only way I can answer is if I know the particular crop you are interested in, and I know what you are interested or what you are looking for in that crop, then I will be able to answer it better. But the word homozygosity or heterozygosity is not something that you can say, okay, is the homozygous that is better or the heterozygous that is better. Because as a farmer, it may be that homozygous that you don't even want, you are not interested in, or the heterozygous behavior that you are just like, what is the meaning of this? And it's that scarce one that is sleeping, that recessive one, that you as a farmer, you are interested in. Okay. So I hope I have answered your question.
Yes, I understand. Thank you so much, ma.
Any other question?
Yes, ma'am. Please, I want to ask question. Okay. This plant breeding of crossing, aside of crossing it with pen and paper using letters, if I want to cross, how do I cross on the real plant? Like if I want maize, can I cross maize? Why cross maize? How?
Yes.
Why another corn, white corn and brown corn together to plant?
Yes, you can do anything you like as a breeder. You can cross yellow, and even if there's blue corn, you can cross it together. All you have to do is to use the pollen. You know this pollen that is on that thing that flies around on the corn. That's the pollen. And then that silk on the female, on the cob, that's the female part of the cob. So you bring the pollen from wherever you like, and then you put it. There's a technique, that one is on the field. When you get to your 400 level, you do some of those things. We call it crossing, pollination. Do it. And even if you want to do your project, if that is what you are interested in, it's a technique. It's not something that you can just do, because you have to be careful so that you will not contaminate your crop. Sometimes pollen flies from very, very far, and then they come and pollinate your crop. So you cover it. There's a way they do it. They cover it with this brown paper. And then when it is time to shed it, there's a way they do it. It's not something that I can describe. You can see there are a lot of videos on YouTube that most of these things, but it's a practical class. It's something that you do on the field. Any other question?
Yes, ma'am. My next question is it possible to cross this with another plant?
Is it possible to
cross like for example, to cross another plant, breed another plant with another one, like beans and maize? Can they go together?
No, it's not possible.
Based on this dry bread that we're having different letters, A, B, 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 C. So what is where are they coming?
Can you cross a cat and a dog together? Eh? Cat has his own physiology. Dog has his own physiology. There are different species of dog. You have Alsatian dog, you have Caucasian, you can cross the different dog together. If they are all dog, cross them together. If they are all cats, cross them together, but you cannot cross cat and dog. So also with crop. There are different types of maize. If you don't know, there are like 5 or 7 types of maize. 5 or 7. You have the flour maize, you have the popcorn, you have the pod corn, you have the common maize that you eat. There are different types of maize.
Even the
You
colors.
want to cross different colors, yes.
Different colors.
You have brown maize, you have white, you have yellow, you have
Purple.
different types. If you want to cross, that's the work of breeders. So if you want to cross maize, cross all the maize together. Now, somebody, you are asking, where did they get all those alleles? You see one maize plant growing. There are so many things that are involved in the growth, so many genes. Like I thought, there are so many. Even you as an individual, even you as you are, you don't know everything in your body except you subject yourself to DNA. If you subject yourself to DNA, you will know everything that is happening in your body. Some of you don't know that you are carrying all manner of things in your body. You don't know. When you give birth to children, you begin to see all manner. You wonder, what is happening to me? So breeding is very interesting, and if you are interested in it, then you can consider it for your second degree. It's interesting. It's very, I don't want to scare you, but it's difficult as well, because you know that you are dealing with so many things that you cannot see. So to you. The absence of any question, I think you can call to be.
Thank you, ma'am.
Your assignment. Your
Thank
assignment
you.
is important.
But
Like
that
I
assignment,
said, I've already set my questions.
the chain for tribe is going to be too long. How do we quickly get it like that?
The what?
The chain outside, the ratio, genital ratio for tribe rate, it's going
It's
to be
not
too
too
long.
long. Try it. Try it. You haven't even started. You are saying it's too long. Try it now. And let me see. Try
Glory,
it. Let me see.
it's just 8 by 8. It's just 8 by 8 by 8.
8 by 8. That's just 64 now. That's not.
Let's do it in the group.
Try it. I've already given you what you did. I gave you the gametes. So just try it out and count. That's all you have to do. Thank you very much. Don't let me take your time. Most of you are at work now. So any question you have, just send it to my email.
Thank you, ma'am.
Very much. And have a nice day. Till next week.
Okay.